In commercial poultry production worldwide, the incidence of infectious bronchitis is of major economic concern. This highly contagious and widespread disease takes its name from the initial respiratory signs, but also affects the urogenital tract. It causes poor performance in broilers and reduces egg production as well as external and internal egg quality in breeders and layers. Additionally, the birds develop a heightened susceptibility to secondary infections that can lead to a higher mortality rate and an increased use of antibiotics. The disease is caused by the infectious bronchitis virus, or IBV, transmitted via the inhalation of droplets through the upper respiratory tract. IBV shows a number of characteristic spikes on its surface. These consist of proteins and play an important role in the onset of infection and the corresponding immune response. After entering the bird's body, IBV quickly reaches its primary targets, the trachea and the bronchi. The upper respiratory tract is lined with mobile hair-like structures, the cilia. Among them are found mucus-producing cells, so-called goblet cells. Together with the mucus, the cilia provide a self-clearing mechanism effective against many invading pathogens. However, in unprotected birds, IBV is able to overcome this natural barrier and binds to the cells on the surface of the trachea. In doing so, it uses its spikes to attach itself to receptors on the cell membrane. Then the virus pushes on into the interior of the cell. Here it multiplies and subsequently infects other cells, which in turn produce many more viruses. As a result of infection, the cells lose their cilia. In this way, the natural barrier against invading pathogens is progressively destroyed, opening the door for secondary infections with other viruses or pathogenic bacteria. The initial binding to the receptors on the cell membrane by means of the spikes on the surface of the IB virus is a decisive step in the onset of infection. If the binding can be prevented, infection will not occur. Timely vaccination is a way to achieve this. It results in a prompt immune response based on local mucosal immunity, which includes the local production of antibodies and their secretion into the mucus. This immune response is able to stop the IB virus from attaching to the cell. Through binding to the spikes, the local antibodies generated by the vaccination prevent infection. Unfortunately, there are many different variants of IBV with differences in their surface proteins or spikes. Highly specific antibodies are able to recognize one type, but usually not others. This is why, despite vaccination, a different type of IBV will be able to infect the birds, causing infectious bronchitis.